So thanks for your patience. Welcome to today's webinar, studying the effects of flow on juvenile salmonids and the stream food. My name is Paul Gannett, and I am manager for Onset's environmental data loggers. And I'm happy to have with us today Gabe Rossi, coastal river ecologist with UC Berkeley and California Trout. This is, if any of you can't hear me, just a little guides here of where to look on the control panel. So hopefully all of you are hearing me. If you have any questions, you can enter them into the questions panel. To open the questions panel, you just click on the triangle next to the word questions here in the gray bar. And you can enter those questions there. We will be saving the questions for the question and answer period at the end of Gabe's discussion. We're going to allow about 20 minutes or so for questions then. So we'll get to them then. We're planning on about 60 minutes in total, and we are recording this webinar, and it will be made available to you afterwards. So during my 25 years with Onset, I've had the pleasure of working with our users and our engineers to develop our lines of HOBO data loggers for outdoor and water environmental monitoring applications. So a lot of the loggers that, for those of you that are users today, most of the loggers that you're using are probably ones that I worked with our engineers to develop. And Gabe is a postdoctoral researcher at the University of California, Berkeley, as well as the coastal river ecologist for California Trout. Gabe studies, he's also the coastal river ecologist at California, Berkeley. Gabe studies the ecology of Pacific salmon with a focus on how juvenile salmon behavior and life history respond to river food webs, stream flow, and physical habitat. Currently, Gabe consults with several environmental flow working groups in California. Over 12 years as an aquatic ecologist, Gabe has worked with state and federal regulators, municipalities, water districts, Native American tribes, agricultural interests, and NGOs to navigate the complex ecological and human factors that influence the recovery of healthy river ecosystems and wild salmon populations. So with no further ado, I am going to turn things over to Gabe. I'll join you again when we get to the question and answer section. So let me just go to my control panel here. I'm not getting that control. Caitlin, can you switch it over to Gabe for us?
Oh.
Sorry about that delay.
All right. No worries. Can everybody see my screen? Through Paul?
Do you have control, Gabe?
I do have control. Can you see my screen, Paul?
I can see it. You're good. Thank you.
I think we're good.
All right. I appreciate it. Thank you. Thank you,
Yes,
Paul, for the introduction. And thanks, everyone, very much for coming today. I took a quick look at the attendee list before the talk, and I was struck by how many folks on the call today are from not only very different parts of the United States and different agencies, but also from different countries around the world. And so I realized that it'd probably be helpful to give a little more context and background to the talk than I had originally intended. So I'm going to do that here. I'm speaking to you today from California, and even though I'm in the northern part of the state, perhaps not many people associate California with the Pacific Northwest. But as many of you, the journalist Timothy Egan wants to define the Pacific Northwest as simply anywhere salmon can get to. And so by that definition, we are the southern extent of the Pacific Northwest because we are in fact the southern extent here of the range of Pacific salmon. And as many of you are probably aware, Pacific salmon have been facing desperate challenges in their recovery over the last century related to climate and harvest and hatchery management and habitat degradation, and especially here in California related to the alteration of natural stream flow in rivers and streams. Salmon are anadromous animals, right? That means the adult fish which live in the ocean enter freshwater rivers and streams where they spawn, and then the juveniles are born there in those freshwater streams, and they can live anywhere from a few months to a few years in those environments before they head back to the ocean. And much of coastal California, where salmon live, is in a Mediterranean climate, which means we get a lot of rain in the wintertime and very little rain in the summer. And the salmon populations that live in California have been beautifully adapted to that climate. And they adapt to it often by spawning and rearing in streams that are really on the physiological knife's edge to what they can endure and what they can sustain. But those streams and that kind of knife's edge physiological environment actually can produce really productive growth of salmon when conditions are suitable. And some of the highest growth rates recorded for juvenile salmon have been in California in these type of streams. Many of these streams are intermittent in the summer, and meaning so surface flow will cease between riffles and pools will become isolated standing pools where fish will continue to rear. Some of these streams go entirely dry, where fish will have to outmigrate to survive. However, with human development, with agricultural development, water use, a lot of these streams, which are, like I said, on that very precarious balance between suitable and unsuitable habitat for fish, have become unsuitable and to the point where fish can't occupy them any further. And so over the past few years, there's been a lot of work, back up. Over the past few decades, there's been a lot of work in California to recover salmon. But what I'm going to talk about today is some work on flow enhancement. And so when I talk about flow enhancement, I'm talking about work where conservation groups and researchers and allies are working to augment stream flow in these intermittent streams from off-channel pond sources. These are often agricultural ponds or municipal water use ponds, and the volumes of water for flow enhancement are much smaller. So when many of us hear the phrase flow enhancement, we might be brought to mind, the large dams and the tail race fisheries that have happened over the last century, where there's large impoundments on a big river and water is perhaps released below the dam to meet both fisheries needs and agricultural needs and human consumption needs, back when we were living under the illusion that we restore salmon with dams. The scale of flow enhancement I'm going to talk about today is very different. These are projects where there's no impoundment. They're on small tributary streams, and the volumes of water are anything that might come out of your garden hose to maybe up to a fire hose. We're talking volumes that are augmented in rates of gallons per minute or liters per second, depending on your metric of choice, and smaller seasons of diversion or augmentation. So this work is designed ideally to offset the anthropogenic effects of water abstraction and habitat diversion, rather than to create some new environment that didn't exist previously. However, while this is a promising, avenue of recovery and research, there has been not a lot of work done studying the effects of this type of flow enhancement, and so that's what we're going to talk about today. I'm going to stop sharing my webcam now because I have to play some videos for you. So I talked about this Mediterranean stream environment. A picture's worth 1,000 words, so I'm going to show you 1,000 pictures, which is worth 1,000,000 words, hopefully. And you can see a time-lapse video here of the decline of stream flow from spring into early fall in a tributary in coastal California. Go to video here. All right, this should play the video. Takes a second to load. So hopefully now you're seeing a time lapse of declining flow. So you can see the stream flow entering from the right side of the screen on the riffle, to the pool on the left side of the screen. And here by about July, that riffle goes dry. So it's a disconnected pool, and flow continues to decline, in this case to the point that the pool evaporates or entirely is lost to groundwater at that point. So that's a quick shot of what those stream environments can look like. Go back to main screen here. So you might ask, why would a fish even want to live in an environment like that? And so from some work we did previously, we looked at the energetic profitability of foraging in these type of streams. And in fact, in early spring and early summer, they can be highly profitable environments for salmon foraging. And of course, not all those pools dry up entirely like the one I showed you there. But a lot of the work that we'll be talking about today, the work oriented towards stream flow enhancement, happens towards the middle and the end of summer and early fall, when you're near that threshold where survival and maintenance of fish through the late summer, early fall period becomes critical. So what I'll talk about today is our work on Porter Creek, the site layout and experimental treatment. I'll talk about how then we observed effects from augmentation on dissolved oxygen, water temperature, riffle crest depth, invertebrate drift, and some effects on juvenile salmonids, particularly their movement between pools and through the stream channel, their growth and survival. You're looking at a picture here of Porter Creek, which is in the Russian River watershed. That's about an hour north of San Francisco in California. And this is the territory of the Pomo people. In fact, Porter Creek was a important pathway between the coast and inland populations of Pomo historically, and it's still very important to the Pomo people who live in Sonoma County today. So the Porter Creek watershed, as I said, it's a Russian River tributary. It's about 20 kilometers squared, and it has that Mediterranean hydrology where many years it's intermittent in late summer. The wettest years it may be connected, and in the very dry years there may be reaches that dry entirely. And it's native habitat to two salmon species, in particular the steelhead trout, Oncorhynchus micus, and coho salmon, Okisich, as well as a host of other native fishes as well. There's sculpin and roach and Sacramento suckers and Sacramento pikemen who all reside in the stream, and then a bunch of amphibians and really cool fauna in this environment. So lower Porter Creek has been developed for vineyard production. This is kind of Sonoma County wine country. And so the first 2.5 kilometers of the stream are pretty heavily developed for vineyard production. And then the upper part of the stream, which goes for another 8+ kilometers, is much less developed and much more wild. The riparian is more natural. There is, some residential homes up there, and there has been historic logging in that area. But in terms of the current landscape, it's much less developed. As you can see, there's a large irrigation reservoir in Porter Creek, which is about 250 acre feet. And that reservoir is used for irrigating the vineyards. But during the 2012 to 2015 period in California, it was one of the driest periods, in fact, the driest period since modern record keeping began in California. And so resource managers looked out to partner with landowners to enter into these voluntary drought agreements where they would engage in activities that would improve stream flow and enhance stream flow on their properties. And in this case, the vineyard owner, Gallo Vineyards, agreed to release water from their reservoir to benefit out-migrating juvenile salmonids as well as over-summering salmonids. And so they developed an augmentation structure. So this is a really cool set of plumbing manifold here with butterfly valves and automated solenoids that will release water at specific times and places in the stream. And this is controlled with a computer control system that allows you to set flow release schedules and monitor volumes that are augmented to the stream, as well as monitor water temperature entering the augmentation. So I'm going to talk about some work that we did in 2019 and 2020, but I want to show you what piqued our interest in this flow enhancement work earlier on. This is some work from 2017, where we were lucky enough to just have data loggers in site for a opportunistic monitoring of these animals. And I was studying fish behavior at the time. We were using some 3D videography to look at foraging behavior of juvenile salmonids. And we were able to capture this 1 week interval where prior to augmentation flow was approaching 0. you can see on the left-hand side, I have stream flow, water temperature, and dissolved oxygen. And I've got 2 red arrows, a before and an after interval. And I'm going to show you video from the before and the after interval of salmon behavior, juvenile salmon behavior. You also can see, if you look at water temperature, that the effect of augmentation here had a very negligible effect on temperature, but a significant effect on dissolved oxygen at the bottom left panel there. Prior to this augmentation, oxygen had dropped down below 4 milligrams per liter, which is entering a stressful range for juvenile salmonids, significantly stressful, and it increased quite a bit after that. So I'm going to play those videos so you can get a sense just visually of what the change in the stream was. So hopefully you're seeing this. This is the before video. There's a single coho salmon that's swimming through this pool. It's near the water surface. It's swimming around the pool volitionally. It's staying high because that's where there's more oxygen in the pool. And you can sorry, I was on mute there. And you can basically see that this fish is not a happy animal in this pool at the time. Now I'll play the after augmentation video. And hopefully you're seeing here, there's a lot more fish in the pool. They're maintaining an upstream orientation. They're drift foraging on prey that's moving through the water column, and it's clearly more profitable environment for fish to be in. So things look good from just a quick look under the water perspective, but let's look closer at the data and see what these effects actually were. So I'll go back to sharing my main screen. There we go. So let's talk about the site layout first and our experimental setup. So the design of the augmentation treatment lend itself really well to a before-after control impact design known as a Baki design, where we had some control study sites upstream of augmentation, and we had what we call impact study sites downstream of augmentation. And so we replicated our study sites on what's called the Riffle pool unit. And this is a classic morphology in streams that have slopes between 3% and 0.5%, where you have a riffle feeding into a pool. It's a natural habitat unit for salmonids as well, because a lot of invertebrate and food production occurs in riffles and drifts into pools where juvenile fish that are living in the pools can take advantage of that food resource and find shelter and habitat in the pools. So we had 2 control reaches upstream and 2 control reaches downstream, and each of our reaches consisted of 4 Riffle pool units. So that was our unit of replication above and below. The Riffle pool units were bounded by pit tag antennas. These are passive integrated transponder antennas. And what those do is they detect any fish that moves from one pool to the next that has a pit tag implanted in it. So we can look at the movement of fish between pools. In each pool, we installed data loggers. So we installed the HOBO U26 dissolved oxygen and water temperature data loggers and the U20 water level data loggers. And we also installed a staff plate to measure water surface and make a correlation between stage and depth. And then we also installed staff plates on the downstream riffle at the riffle crest to make a correlation between depth over the riffle and continuous stage change in the pool. We also measured the drift of invertebrate prey from riffles into each of these pools and velocity along the entrance of the pool, which is an important metric for where fish are foraging. So looking a little closer at the site layout, this is just the individual riffle pool unit. You can see those pit tag antennas on the left and right bounding the unit. Flow is moving from left to right in this picture. You can see the drift net at the head of the pool and our cross sections for measuring pool hydraulics. And then the Riffle Crest Thalwag location, RCT, down here at the bottom of the pool. Looking at the profile view on the bottom of the screen, you can see where we installed the data loggers. And we surveyed using auto level the elevation of the data loggers relative to the elevation of the downstream Riffle Crest. And what was great about that was it allowed us to convert stage in the pool to continuous stage in the Riffle Crest. So this setup gave us 16 pools, 8 above and 8 below, with data loggers in each one of them recording data on a 15-minute interval. So we were recording thousands and thousands of data points every day, which we did all throughout the course of the summer. It gave us a really rich data set to compare against the behavior, survival, growth, and movement of the salmonids, as well as some of the stream food web data. There was a question earlier about how we mounted our data loggers. I'm not sure if we did it the way according to exact company profile, but what we did was we created these vented PVC housings that were 2-inch housings that the HOBO loggers could fit inside of. There was holes drilled in them, and then we used beefy zip ties to hold the loggers in place within the housings. And then we pounded 5/8-inch rebar into the bed, and we used hose clamps to attach the housings to the rebar. And these were all surveyed in relative to the locations of the other instruments that we were using. One word of caution I learned was, this is a freshwater environment. I did some work in a saltwater environment, and those hose clamps, which are stainless steel hose clamps, do not have stainless steel pins. And so while the clamp itself will not rust in a saline environment, the pin will rust relatively rapidly. So make sure you get hose clamps with stainless steel pins if you're working in a saline environment. Okay, so looking a little more closely at our study over the last 4 years, we worked from 2017 to 2020 on this work, and it allowed us to look at a wide range of summer hydrologies in the stream. So this is a hydrograph with daily stream flow on the left axis and date on the Y. And because there's a lot of work to talk about here, I'm going to focus on 2 very different summers, on the wet summer of 2019 and the very dry summer of 2020. And in both summers, we had a similar augmentation treatment, a 1-month duration of 14 liters per second, which we tried to initiate just prior to disconnectivity. And so comparing this very wet summer and this very dry summer will allow us to look at the effects of augmentation on salmonids and the stream food web under different ambient natural conditions. So here again is the hydrographs, and I've highlighted the period of augmentation in both of those years. You can see the extent of augmentation was longer in 2019, but we're only going to focus on the 1-month period. All right, we'll start with dissolved oxygen. As I'm sure many of you know, dissolved oxygen is a really critical currency for all freshwater fish and all aquatic animals, for that matter, that are living underwater. For our salmonids, it's a really important currency in their base metabolic rate, in their prey assimilation efficiency, in their swimming performance, and a whole host of physiological responses that allow them to survive and grow in these streams. So it's a really good indicator of the suitability of habitat for salmonids. So rather than show you data from all 16 of those data loggers, I cherry-picked a few sites, both in the augmented reach on the left side of your screen and in the control reach on the right side of your screen. And I separated the years. So 2019, remember that's the wet summer, is on top, and 2020, that's the dry summer, is on the bottom. And then I highlighted the period of augmentation in yellow here on the left side. So what you can see just visually without any statistical analysis is that augmentation during the wetter summer had a much smaller effect on dissolved oxygen, and that's because the ambient conditions were relatively suitable throughout the early summer and high enough that our treatment wasn't able to have a significant effect on it. Although you will see that there was a significant effect in diurnal variation in dissolved oxygen during 2019 at one of our sites. However, in 2020, you can see that dissolved oxygen had decreased to the point where it was near zero, in fact, which is of course lethal for salmonids. And our 1-month treatment of augmentation was able to increase oxygen to between 4 and 7 milligrams per liter with a significant diurnal flux in these sites. And that was consistent across all of our study sites. There was variation in the amount, but the statistical analysis showed there was a large and significant effect on dissolved oxygen in the treatment reach. Now, you might be asking, what's going on with this really wonderful oxygen that happens in September and October? Did the stream miraculously get a lot better? And no, that's actually what happens when your data logger is exposed to the air and the stream is dried entirely in that case. So these are data loggers that ultimately ended up, the pools became so low that those loggers were exposed to the air, and that's the oxygen of air being measured. I have two thresholds written on here as well. I forgot to mention the dashed red line is considered negligible impairment for dissolved oxygen at around 7 milligrams per liter, and the solid red line is where you start to see significant mortality. I always put threshold values with a little bit of trepidation or a grain of salt, because of course there's a whole host of co-varying ecological and physiological variables that really mediate what those thresholds mean to any individual animal. But these are good general numbers to start thinking about as a introductory way to think about the effects of augmentation on dissolved oxygen. So let's take a look at water temperature next, and here's a little Riffle sculpin from Porter Creek. You can see here the same design in the charts: augmented sites on the left, control sites on the right. 2019, the wet summer on top; 2020, the dry summer on the bottom. You can see, if you look just visually, that the effect of augmentation on water temperature was much smaller than the effect of augmentation on dissolved oxygen. And that was consistent across almost all of our sites and across both years. This might be surprising to some people because we hear, in stream ecology, oh, there's a strong correlation between dissolved oxygen and water temperature. And it is true that, colder water can hold more oxygen, but that isn't necessarily giving credence to the causes and mechanisms that actually create dissolved oxygen dynamics in streams, right? There's aeration, there's biological consumption of water, there's photosynthesis and the production of oxygen that way, so bacterial respiration. There's a whole bunch of physical and biological factors that mediate the dissolved oxygen dynamic besides water temperature, and they're not all necessarily directly a function of water temperature. And so those things, while they're correlated, aren't necessarily causal. But you can see, especially in 2020, if you look at the bottom two sites here, two very interesting differences. In one of our sites, we found that augmentation reduced diurnal fluctuation in temperature. In the other site, it actually increased diurnal fluctuation in temperature. And we think this is because in some of the sites, there were very shallow, wide riffles where increasing the flow allowed for warming and cooling of the water throughout the day due to ambient temperature dynamics. And in others, they were very shaded, or flow was moving through cobbles and gravels, and those fluctuations were muted. However, statistically, there were very little treatment effects from augmentation on water temperature in this study. So now I'll talk about riffle crest depth briefly. Riffle crest depth is a really important hydraulic measurement in these type of streams. So the riffle crest is the downstream extent of a pool, and it acts like a natural weir. So it functions almost like Mother Nature's weir, and it has a strong effect on hydraulic and water quality and physiological conditions upstream of that weir. And we've got a publication that was just accepted on this relationship between riffle crest and the weir and its potential uses in stream ecology and management. There was some really wonderful work by CDFW between 2017 and 2020 looking at the relationship between riffle crest depth and dissolved oxygen in the downstream pool. And they found a strong correlation across 8 different streams in the San Francisco Bay Area, where as you start to drop below 6 or 7 centimeters, you see a strong decrease in dissolved oxygen. And digging into the mechanisms of this is kind of beyond the scope of my talk. It's probably a very complex set of mechanisms, but a lot of it has to do probably with turnover rate and residence time of water in the where it's interacting with biological agents in the stream. So at higher flows, there's lower residence time and less interaction between the moving water and the biological agents that affect dissolved oxygen. So we took a look at riffle crest depth in both of our streams, and this is the same design again. And you can see that in the wet summer of 2019, when stage remained connected all summer and water was flowing well all summer, we had a negligible and statistically insignificant effect on riffle crest depth. But in the summer of 2020, we had a large effect on riffle crest depth, where it went from near 0 up to 5 to 7 centimeters in some of our pools, as you can see here on the bottom 2 panels. And you can look at the natural recession of riffle depth here on the right side, where in 2020 it decreased, or 2019, sorry, it decreased but never became disconnected. And in 2020, in almost all of our sites, it became disconnected by early August. So this is another promising finding from the data loggers. This is another, data loggers don't lie to you, and this is something I learned. We saw this small increase in stage that I've circled here on the bottom left of the series, and I was scratching my head why we didn't increase any water. There was no rain event that occurred during that time. Why did we have an increase in stage? And I realized it was when we had actually put block nets into the stream to do some fish sampling, and leaves had built up on the block nets and actually increased the stage in the pool for those few days. So it was an artificial artifact of our experimental work. So I actually had to remove those days for our statistical analysis, but it's a good lesson in that data loggers aren't lying to you. Most of the time, I hope. So let's take a look at invertebrate drift. As we discussed, these riffles that invertebrates are produced all throughout the stream, and they're a really important and amazing and fascinating part of the stream food web. But for the perspective of thinking about salmon foraging and fitness, a lot of this work focused on the drift of invertebrates from riffles into pools and the ability of salmon to take advantage of that drift. And I'll be relatively brief with this. What we found was that in both years, the impact reach, where we released water, the augmented reach, had a lower decline in drift during our study than the control reach, which is positive. However, in 2019, that decline was, and the variability in the sites was large enough that the statistical analysis was not significant, whereas in 2020, most of the control reach went entirely dry, and we maintained drift in the impact reach, so we did have a significant effect then. However, I think we should take this with a bit of a grain of salt, because if you look at the phenology of drift in this stream from early spring into late summer, while we did show a statistical effect, an increase in the rate of drift, the rate of the background ambient rate of drift is so much lower in late summer than it is in spring and early summer when fish are growing in a positive way, that this change may have had a negligible effect on the overall fitness of fish in terms of food production. I think more work is certainly needed on providing context for that. Oops, went a little too fast there. So let's take a look at our salmonids. I'll look at three components of our salmonid monitoring, both the effect of augmentation on movement between pools, the effect of augmentation on growth and survival. So here we are electrofishing in Porter Creek to capture salmonids, to fit them with pit tags. We would bound each of our riffle pool units with block nets, capture the fish, tag them, release them, and we would use that data for both our growth estimates and our survival and movement work. So with movement, we had two kind of primary questions. The first one was just simply, how does this interpool movement of summer earring, steelhead, and coho respond to flow augmentation? And the second one was, what abiotic variables were strong controls on this movement? The movement of fish within reaches, between pools, without leaving the stream, but within reaches, is really important for them to be able to deal with density-dependent limitations in the pools they're in, to be able to move from pools that become inhospitable to hospitable. It's related to a lot of ecological theory, like the ideal free theory. I'm not going to get into that, but it's a really important variable in the fitness and survival of summer earring salmonids. So because of our experimental design, and because the data loggers were installed in each of these pools, and we had the pit tag antennas bounding them, we were then able to correlate the movement of each detection. Well, I say movement, I mean detection of a pit tagged animal at a riffle with the water quality and hydraulic conditions in the upstream and downstream pool. And so that allowed us to do some really neat analysis and look at a fine-grained resolution on movement decisions that fish made, and we'll talk about that. But first, I'll talk about just the overall response of movement to augmentation. The first thing to notice here, these are all of our detections for steelhead on the top and coho salmon on the bottom. The years are separated. The wet summer is blue, the dry summer is yellow, and then the control reaches where we were augmenting was on the left and the impact reaches were on the right. And the first thing to notice is the ambient background stream flow has augmentation on the movement phenology of these animals. You can see much more movement in 2019 than in 2020. We had almost 100,000 detections that year and much fewer in 2020. And you can also see a strong decline in movement as flows become lower, which makes sense. Interestingly, the decline in movement was different for these two species, and steelhead, Oncorhynchus micus, seemed to have a stronger response to declining flow in their movement patterns than the coho salmon did. So when we look simply at the effect of movement on the effect of augmentation on movement, we found differences again between the species. So steelhead trout had a positive response to augmentation, meaning they moved more, or the decline of movement was less when we augmented flow in the impact reach relative to the control reach. So we had a statistically significant effect on movement in both years for steelhead. But for coho salmon, we did not have a statistically significant effect on movement, certainly not in 2019, and it's very marginal in 2020, kind of on the boundaries of significant, and the effect size was smaller for coho salmon. So this kind of gets at some of the behavioral and physiological differences of these two species. Also, a lot of the steelhead trout were wild native fish, and some of the coho salmon here that were tagged, many of them were hatchery origin fish. So there could be some local adaptation as well. But that's kind of beyond the scope of the talk here. Okay, so looking closer now at the physiological variables that controlled movement. So we created a variable that we're calling usage here. It's on the y-axis, and it's going to take a second to explain, but hopefully it'll be useful to think about how fish respond, how movement of fish responds to these variables. So the way we defined usage was we took the proportion of all the detections that we observed, and we subtracted it from the proportion of time that a certain environmental condition occurred. Okay, so to break that down, on the x-axis, you have riffle crest depth. So if you take our whole study and you pick any depth, say 5 centimeters or 4 centimeters, and you say the proportion of, if you take the proportion of total detections that occurred at 5 centimeters and subtract that from the proportion of time that 5 centimeters itself occurred, you can get this metric called usage. And if you think about that, if the value of usage is 0 on the y-axis, that means that fish are moving at the exact same proportion that environmental condition occurred. If it's a positive value, that means fish are moving more frequently than that condition occurred. And if it's a negative value, they're moving less frequently than that condition occurred. We think this is a valuable metric because it may get at some of the volitional behavior and the choice to move towards or against different patterns. So in 2019, we got 15,000 detections of salmonids that were associated, in this case, Oncorhynchus micus, that was associated with a specific RCT depth. And in 2020, we have 5,294 detections, so quite a few less. But what was striking to me was in both these very different hydraulic years, the increase in movement occurred at nearly the same depths. So we see when we reach stream riffle crest depth of around 4 centimeters, a positive association towards increased movement, and then a decrease in movement below that. There's also some interesting patterns that happen at very deep depths where movement declines again. This may be because at those deeper depths, 17, 18 plus centimeters, conditions in the pools are suitable. Fish don't need to move in and out of them, but we need to do more work to address some of that conjecture. But this lower end of the series here, down around 4, 3, 5 centimeters, shows some strong and potentially physiological limitations to movement decisions for salmonids. So we used the same variable here to look at movement associated with dissolved oxygen. And we saw, again, an interesting, somewhat similar pattern in when fish moved relative to different oxygen values. Again, you see an increase in movement with dissolved oxygen associated with 6, 7, and 8 milligrams per liter in both the wet and dry summers. And what I found striking about that was that those are dissolved oxygen levels that are much higher than maintenance needs for these animals, right? That the fish can survive at lower dissolved oxygen levels than that. But it kind of points to the fact that movement is energetically costly, and that fish maybe don't necessarily feel inclined to move until dissolved oxygen was higher. There is some movement that happens at very low oxygen, maybe in a desperation mode as fish are trying to get from one site to the other as well. All right, we're keeping an eye on my time here. I think we're doing okay. I'm going to look briefly at salmonid growth. And here we have two intervals where we captured fish and measured their growth, one prior to and one after augmentation. What we found though was that we didn't have, let me rephrase that. We don't have a full growth interval before and after augmentation. We had growth interval in the control and the treatment reach. And so we can't actually statistically evaluate the effect of augmentation on growth in a robust way. But what we did find was late summer, right, in the July to August period is a period of very low growth in these streams anyway. And that's what we saw across the board. There was near zero growth for both young of year, that's yoi, so that's the fish that were born that year and are usually less than 80, 75 millimeters long, and older, one-year-old and two-year-old juvenile fish. All experienced nearly zero growth, with the exception of steelhead young of year, which in the augmented reach grew significantly higher than in the control reach. Now, again, since we don't have a pre and post interval, we definitively can say that this is due to augmentation. But when you put this together with the improved physiological environments, that improved oxygen and potentially drift of prey, it may have something to do with, and likely has something to do with the effect of augmentation. So finally, I'll talk about salmonid survival, and we're still working with this data right now, but we really have one year that we've completed. So we did have a pre-survival interval and a post-survival interval where we measured survival over a period of time before augmentation and after augmentation in both years. And we used a robust design from a MARC recapture model in the program MARC to estimate survival at the reach scale. A lot of this work was from work by California Sea Grant and Mariska Obozinski, who's been leading a lot of the research on Porter Creek. I need to mention them, but I'll talk to them about that group at the end here. So what we noticed was in 2020 that we took the difference in survival before augmentation and after augmentation, and we found that for wild and hatchery coho as well as wild steelhead, there was between a 23 and 30% increase in survival in the augmented reach during the dry year of 2020. Now, we haven't worked that up for 2019. It's a very positive finding that we're excited about. We expect that in 2019, because it was a wetter year, ambient survival was probably higher. This effect will probably be less, but we don't know yet. So we're excited to look that up. So what are some of the take-home messages? There's a lot of words on this screen. I'm going to talk through it a little bit. One of the first take-home messages from this study was that the effect of augmentation was, of course, influenced by the antecedent flow conditions, as well as by channel characteristics. I didn't talk a lot about this, but in reaches where we had high phoretic exchange and lots of loss to groundwater, we had a much smaller effect on surface flow characteristics on the variables that I measured here. So it may be that there's a bit of a Goldilocks zone, right? A bit of a hydrologic zone where augmentation is able to have a larger or less or a smaller effect on these ecological variables. We found that this treatment of 14 liters per second produced large and significant effects on riffle crest depth and dissolved oxygen at sites up to a kilometer and a half downstream from the point of augmentation in that dry summer, and small but still significant effects during the wet summer of 2019. However, the effect on water temperature was not significant. Sorry, I need a little water myself. Was not significant in either year. And then we found that there was a significant effect during the dry summer of 2020 on invertebrate drift, but no effect during the wet summer of 2019. However, as I mentioned, we should take that with a grain of salt because the effect size was small relative to potentially what's necessary for a bioenergetically profitable environment. The detection of fish, the decision of fish to move between pools was more affected by antecedent conditions than by augmentation. However, we did see a statistical increase in movement of steelhead, juvenile steelhead, in both 2019 and 2020 from augmentation. And we're kind of excited about this usage characteristic, which suggests there may be a similar preference for movement under certain dissolved oxygen and Riffle crest depth conditions across years, regardless of the hydraulic environment, although that statement does need more research to think more about. And we think this usage variable may be valuable for thinking about management thresholds for abiotic variables on fish response. And finally, during the dry summer of 2020, we found that there was increased survival at the reach scale for Omicus in the augmented reach, and that's encouraging and work that we hope to follow up on. So all this work is currently in revision. We'll hopefully be submitting it soon, resubmitting the paper soon, and look forward to getting that out into a peer-reviewed journal. So I wanted to step back for a second and point out we did this study on the effect of augmentation from off-channel ponds on ecological response in these small streams, but it's not entirely a conclusive endorsement or not endorsement on that this is a management strategy that's going to be highly effective, right? There's clearly some very positive take-home messages from what we learned. The increase in survival is extremely positive. The effect on dissolved oxygen and Riffle crest depth is extremely positive, and I think those are strong reasons to continue this work, and that especially given the decline of fish that we're facing now, that this is an important tool in the toolbox for people working on recovery. But we should also make sure we're paying attention to the stream's natural background patterns and phenology of drift and salmon life history. Again, as I mentioned, these volumes of augmentation shouldn't be so large that we're actually creating a new and different environment in the stream, but rather perhaps that we're making up for some of the anthropogenic effects of water diversion and promoting survival across very dry years. We also need more work on how these treatments affect non-target species. We have foothill yellow-legged frogs, salamanders, and other invertebrates in this stream that we did not study. We should think carefully about the full life cycle response rather than just local survival and improvement, and thinking and placing these kind of flow enhancement work at the watershed scale. How would this stream have functioned historically, and how would flow enhancement support that? So, kind of in brief summary, I feel very positive about the potential for this scale of flow enhancement to be an important tool in the recovery toolbox. I don't think it's a panacea, but I think there's a lot more work that can be done to figure out how and when to use it in an effective way to help recover salmon in California. So with that, I'd like to acknowledge a bunch of folks who are really critical in supporting this work. Obviously, the landowner, Ian J. Gallo, and I mentioned California Sea Grant, Mariska Obuzinski's team in Sonoma County, Sarah Nossman-Pierce and Will Boucher and others that worked with us on this project they invited us into this watershed. They'd already been doing research there. They're really instrumental in the recovery of fish in the Russian River. The COA broodstock program, which was the source of the hatchery fish that we studied here, and some funding that we got from USGS via ANR. Also, Trout Unlimited performed the gauging and hydrologic analysis of the watershed, and they were a great partner. And the Sonoma RCD, who's been working on this stream as well. So with that, I'll take any questions we have and maybe turn it over to Paul to direct the questions to me. Thanks, you all.
And thank you, Gabe. That's been a great presentation of your work and what you've learned. Important work. We obviously are dealing with less and less water, and how to manage that most effectively is just so critical. So thank you. So yeah, I have a few questions here. We have some questions that were sent in advance, and some that I've been getting during your talk. So let me start with one that was sent in advance. This is a question from Dana, and that is, are you using temperature monitors in the hyperheic zone as part of the monitoring program?
Yeah, that's a great question. We did not monitor temperature in the hyperheic zone. We monitored water temperature in pools, and that's a really important question. We know from other work that in larger rivers, when water goes subsurface and moves through the hyperheic zone, you can have cooling of water. That's very important. Whether or not that occurs in these smaller streams, I think needs a little more work, because there's also a lot of biological processing that happens, bacterial decomposition that happens in the hyperheic zone. There's buried carbon in those riffles. So there's a lot of really interesting and important work to think about the exchange of water through the hypereoic zone and its effect on surface flow in the fish that are in pools. Yeah, but we did not measure it directly.
Yep. All right, thanks. Another question is actually related to temperature. This one comes from Steve, and he was asking what was the difference in temperature between the source of augmented flow, the off-channel ponds, and the receiving waters, and how was this controlled or modulated?
Yeah, so we did measure temperature in the augmentation, and I didn't report any of the temperature values there at the point of augmentation. They were relatively close to the background values of the stream. A few times they were several degrees warmer, and we found, as I noted, that as water moved in the channel over 0.5 kilometer to 1 kilometer downstream, that they would normalize to the ambient temperature of the air. Probably because in this case, the volumes of augmentation were very low, and the processes that affect warming in the stream were able to overcome whatever boundary condition that the water started at. But we should certainly make sure that other folks doing this work don't assume, based on our findings here locally, that there won't be an effect on water temperature. That's a function of many things, including the boundary condition of the water and the volume of the augmented flow.
All right, thanks. Got a question from Brandine. Do off-stream flow enhancements affect groundwater flow and/or downstream basin recharge?
Yeah, that's a great question, and something we would like to know more about. We did not monitor downstream groundwater levels as a function of augmentation, but we did certainly find that downstream groundwater levels have an effect on the augmentation success, right? So in times where the groundwater level was near to the hypereoic zone and near to the stream, we would have a much larger effect on surface flow dynamics and water quality in the stream. And obviously when groundwater receded down a lot, we'd have hypereoic loss of our small volumes of water, and they weren't necessarily able to keep up with that loss at lower levels of groundwater. But the relationship, I think this is one of the most important actually variables to understand where and when this type of augmentation work could be effective, is correlating it to groundwater levels. I think it's a really important metric and indicator of whether or not those augmentations will be successful.
Thanks. Here's one that's maybe as much of a comment or as it is a question. It comes from Harvey. You may, I got to get the words in the right order here. You may have moved this type of watershed stewardship from the rear of management to the forefront, meaning an environmental management plan rather than compliance level, urban and agricultural water management plan. In this case, knowing when to give back rather than to take. Does that kind of make sense? And you want to address that?
Yeah, I mean, I think that's a good way to think about it. And yeah, it's a complicated issue, right? Because I think there are, yes, we want to make sure that we're minimizing and mitigating our impacts, and there are things that are positive that we can physically do to, the word enhance is a tricky word, right? Because in one hand, it can be like improving on nature, which makes me very uncomfortable. We're not trying to create and improve on nature or make something, new that wasn't there. But we are trying to give back in the sense of replacing what we have taken and allowing the streams to function in a way that they would have otherwise, that they're not currently allowed to function. And that's more than just simply replacing the exact volume of water that was taken. We have to remember these streams are impaired also geomorphologically in the riparian zone, in many different characteristics. So the relationship between flow and ecological response is not so simple as simply gallons to gallons, right? So yeah, I'll stop there because that's a whole other seminar probably.
Yeah. Speaking of other seminars, there was the question on deployment tips for mounting data loggers. I just wanted to mention that for those of you who are looking for suggestions on how to mount data loggers, you showed how you mounted your loggers. I wanted to just mention that if you go to the Onset website, we do also have a webinar on deployment tips for HOBO water level loggers. So if any of you are looking for more ideas on mounting, you can refer to that. So just, let's see. Oh, we're starting to get some fairly good number of questions here. I did have a request from, where is it? It's not scrolling up. To share a web link for somebody who's also working in the Porter Creek area. Let me, I'm sorry, I'm trying to scroll up to that.
There's questions or requests for data and things like that that we can't cover here. You know, I think my email is available to folks, and you can send me, you know, questions or requests, and I'll try to address those as well, and get what we can share to you when it's available.
Yeah. There's actually some more in-depth questions in here that I think we'll have to follow up on. So
yeah, I'd be happy to do that.
Yeah. The actually, if you could pass control back to me, because we're starting to get down to the final bits of time, I'll show our content information slide. Let me.
I think you should have control now.
Hold on.
Yeah.
Just give me a minute here. I need to move the, there we go. Thanks for your patience. I think the more attendees we have at these webinars, the slower the response is. So it's good that we have many of you attending today, but we're pushing the limits of GoToWebinar. So this slide, you're probably seeing my screen now, but I'm going to show it in full screen mode. So yeah, the link that I'm referring to was from somebody with the NMFS, who was also doing some work in the Porter Creek. This is from Daniel. And Let me just share. You probably know him, but he asked me to share this link with you, with the attendees. So I'm going to copy this over to the chat. And well, I'll copy that in a minute, but sorry. My computer is really slow to respond here.
There we go.
My apologies, everyone. I just wanted to get a couple more questions. Let's see. So yeah, we've got a request for links to white papers related to this. Yeah, we'll send out some links to related white papers that we have at Onset, and Gabe, you're willing to or welcome to add to that. Will this presentation be available for review later? Yes, we'll send out a link to the webinar recording afterwards, so you'll all be able to have a copy of that. Yeah, I guess here's a question from Zach. Was there any consideration of non-natural equipment presence influencing fish movements, or was this factored into the control reaches?
Yeah, that was a great question. I mean, we really instrumented this stream, and it's something that we were concerned about and thought a lot about. In terms of fish movement, the antennas were mounted above the water surface, so they didn't affect movement. Whether or not shadow had an effect is debatable, but probably negligible. In the stream itself, the only instrumentation that would have potentially affected movement was the rebar and the data loggers, again, which didn't really have probably a significant effect on movement. Now, we did, of course, net the stream when we were electrofishing, and we removed those nets. And the day before and the day after, fish were stimulated to move in probably an unnatural way. So we had to consider removing that data before we evaluated them kind of going back to a background behavioral state. Yeah, no, that's a great question. And when you have this level of instrumentation in a channel, you really do have to think about whether or not you're creating an artifact that's affecting your ability to understand the response.
Good, makes sense. So we've hit our 1 hour time, so I think we'll cut questions off at this point. But as we said, there's several more good questions in here, and some of them are product specific, so we'll follow up with those, more from the Onset side, and some are more application based. And hopefully, Gabe, you can we'll find the time to answer those questions as well. So we'll do our best to answer all your questions. So so Gabe, I just really want to thank you for sharing your insights with us today, and I want to thank all of you who have been attending for joining us today. Is there any closing remarks you'd like to add, Gabe, before we
no, it was a pleasure. Yeah, I was happy to give the talk, and always happy to talk about this work and other work. And, like I said, my email's there. Please don't hesitate to reach out, and I'll do my best to get back to you. And good luck with all your work. Thank you, everyone.
All right, thanks.